Hello, everyone, and welcome to Chapter 14 on antimicrobial drugs. In this chapter, we will explore how various drugs are used to combat microbial infections. We will examine the different types of antimicrobial agents, their mechanisms of action, and the growing concern of drug resistance. By the end of this presentation, you should have a stronger understanding of how these drugs work and the challenges associated with their use. To start, let's clarify some key terms. Chemotherapy refers to the treatment of diseases using chemicals. And so a chemotherapeutic agent or drug is any chemical used for disease treatment. And antimicrobial drugs specifically target microorganisms, helping to inhibit their growth. And these drugs can be classified based on their source and method of production. There are three main types of antimicrobial agents. There are natural antibiotics derived from living organisms, such as penicillin from fungi. And then you have synthetic drugs. These are created artificially in a lab, such as sulfa drugs. And then finally, a mix of both, you have semi-synthetic drugs. These are chemically modified versions of natural antibiotics, like methicillin. This table here shows some of our naturally derived antibiotics, the microorganism in the left column, the type of microorganism that is from the center column, and then to the right column, the antibiotic that was derived from that particular species. Something you'll notice here is, interestingly, over 50% of known antibiotics, natural antibiotics, originate from a single bacterial genus. That's called Streptomyces. The Streptomyces is a common soil-dwelling bacterium, and as such, it is surrounded by other microbes, and they needed to compete for limited resources effectively against those other microbes. So therefore, this particular genus, it got really good at producing a wide variety of chemical weapons, so to speak, or antibiotics, in order to combat them. And we have a pretty long history now of antibiotics that have come onto the market since about the early 1900s. The period, however, from the 1990s onward is often referred to as the antibiotic discovery void, because during this period there was a significant decline in the discovery of new classes of antibiotics. This era saw pharmaceutical companies shifting focus away from antibiotic research due to economic incentives, regulatory changes, and the misconception that bacterial infections were largely under control. And consequently, the pipeline for new antibiotics has been quite thin, and this exacerbates the global challenge of antibiotic resistance. There is a renewed interest in antibiotics. For example, this particular antibiotic called Teixobactin is a promising new antibiotic that is derived from soil bacteria, no surprise. It has shown effectiveness against some drug-resistant bacterial strains and offers a potential solution to increasing problems of antimicrobial resistance. This particular drug attacks the cell wall synthesis, so I believe it's peptidoglycan, like a lot of our antibiotics do. One of the most critical properties of antimicrobial drugs is selective toxicity, the ability to target microbes while sparing human cells. And this is achieved by targeting microbial structures that humans either lack or have different versions of. And a great example of that is the ribosomes. Both prokaryotes and eukaryotes have ribosomes, the protein synthesis machinery of the cell. However, as you have learned in the past, they are a little bit different from one another. Namely, we learned that prokaryotic ribosomes were about 70 Svedberg units in size. Again, that's how quickly they form in a sediment layer if you were to spin down the intracellular contents of cells. Um, this number represents like how well gravity pulls on those ribosomes and has them settle out. It's a way of measuring basically the mass of different intracellular products. And eukaryotic ribosomes are slightly larger. There's no surprise there. And that's roughly 80S. What we haven't discussed is some of the constituent parts of a ribosome. Now, you see in this image here, there are different RNAs that are listed, and that is because ribosomes are, yes, they are able of conducting enzymatic activity. That means they're protein catalysts. They also have RNA in them as well. So there's proteins and RNA that build up the structure of a ribosome. And in this lecture, we're less focused on the ribosomal RNA, but we're really focused in on the protein subunits. There are two subunits of prokaryotic ribosomes, a 50S subunit and a 30S subunit. And for eukaryotic ribosomes, that's a 60S subunit and a 40S subunit. Now, we'll be looking at antibiotics that aren't active against human ribosomes. So we're going to be more focused on antibiotics that somehow interact with either the 50S or the 30S subunit. So keep that in mind as we move forward. And sometimes this 30S subunit is referred to as the small subunit, and the 50S subunit is referred to as a large subunit of the ribosome. There are other common targets, and these include the cell wall. Bacteria have them, but humans do not. Ribosomes, as we've already discussed. And then, obviously, prokaryotes have different metabolic capabilities and different metabolic pathways that can be targeted as well. One of those is going to be the synthesis of a cofactor called folic acid. Antibiotics are classified based on their spectrum of activity. A narrow spectrum antibiotic targets specific bacteria. Examples include polymyxins that are effective only against gram-negative species. Broad spectrum antibiotics target a wide range of bacteria, including both gram-positive and gram-negative species. An example of this is tetracycline. Broad spectrum antibiotics can be useful, but may contribute to antibiotic resistance and superinfections. This image here shows the spectrum activity of many different common antibiotics. As you can see, the tetracyclines cover both gram-negative, gram-positive, and even some other organisms that are outside of just the general gram-negative and gram-positive categories that are hard to treat, like chlamydias and rickettsias, which we'll talk about a little bit later in this lecture. As you can see, some of these narrow spectrum antibiotics, such as polymyxins, that only attack gram-negative bacteria, and streptomycin, another one of those from Streptomyces that can attack both the mycobacteria, which are tricky to treat due to their acid, mycolic acids, and gram-negative bacterium as well. Sometimes antibiotics can cause something called a superinfection. A superinfection occurs when a broad-spectrum antibiotic disrupts the normal microbiome. This allows for the repopulation with an opportunistic pathogen, which then is able to thrive without serious competition from others. This can lead to a secondary infection and is often harder to treat than the initial infection. Dosage refers to how much of a drug is administered over a certain time period. It is determined by factors such as the patient's age and body mass, the half-life of the drug, in other words, how long it stays active in the body, and individual patient considerations. The goal is to maximize effectiveness against microbes while minimizing the toxicity for the patient. Thereby, we want to prescribe what we call the optimal dose. That's kind of right in the middle of this range. Antimicrobials can be administered in different ways. They can be administered orally through tablets or capsules, and this is convenient, but may result in lower plasma drug levels and longer times until the maximum absorption is reached. If you look at this graph here, this is measuring the concentration of the drug in a patient's plasma over time, and time t0 is when the patient is first exposed to the drug. And we can see that the oral dose has the longest lag. So you need to digest this drug, it needs to be absorbed, and it's just that process, not as much of it is necessarily going to reach the plasma or the blood. On the other hand, intravenous or IV or intramuscular IM result in faster and also results in higher plasma drug levels. And this is often used to treat severe infections. Some drugs are ineffective when taken orally too, due to poor absorption in the digestive tract. We're going to be taking a look at a lot of classes of antimicrobials. Antimicrobial drugs are categorized based on the type of pathogen they target. They may be antibacterial drugs that target bacteria, antifungal drugs that target fungi. They might be antiprotozoan or antihelminthic, which target parasitic worms. Each class has unique mechanisms to inhibit or kill the pathogen. This again gets to the different targets or mechanism of action of antibacterial drugs. There are 5 mechanisms of action for antibacterial drugs. Some antibiotics, such as penicillin and cephalosporins, work by inhibiting bacterial cell wall synthesis. These drugs are especially effective against gram-positive bacteria. Some, like polymyxin, disrupt the cell membrane, while others may interfere with protein synthesis, such as tetracycline or streptomycin. Others may interfere with specific bacterial metabolites. And lastly, they may interfere with nucleic acid synthesis. We'll unpack each group one by one, explain their mode of action, and important antibiotics belonging to each category. And so this chapter will therefore require students to be effective at identifying many types of antibiotics. Let's review the basic structure of peptidoglycan so we can understand how it is targeted. Peptidoglycan is made up of proteins and sugars, hence the name peptide and glycan, meaning short chains of amino acids, and glycan meaning sugars. The two sugars are abbreviated as NAM and NAG, but this stands for N-acetylglucosamine and N-acetylmuramic acid. And as you can see in this image here, it shows the difference between gram-positive bacteria and gram-negative bacteria. We'll dive into that little detail here just to help you focus on the structure here. And so as you can see, peptidoglycan, the sugars are alternating sugars between NAM and NAG. And that is in common with both gram-positive and gram-negative bacteria. And the peptides come in handy when it comes to bridging these chains, these polysaccharides together. See, in order to connect one older layer to one newer layer, a series of amino acids, represented by these colored balls here, are used to cross bridge between one chain of polysaccharide to another. And in gram-negative bacteria, these short peptides are directly linked to each other. So the alanine directly binds with the lysine. However, for added strength, the gram-positive bacterium actually use another, what's called a pentaglycine cross-link. Glycine is just some amino acids, and penta means 5. So there's these 5 amino acids that help link these two amino acids together to make a stronger bond. Now, that's not detail that you need to memorize, but you should be aware of peptidoglycan being made of chains of polysaccharides that are linked together through these peptide bridging processes, and that allows them to cross-link these different layers together. So we'll begin by discussing the most common bacterial target of antibiotics, and that is the cell wall. This was the target of the first antibiotic discovered by Alexander Fleming. We discussed in chapter 1 how he left on a trip, and upon his return he noticed that there was a fungus growing on one of his old agar plates. And where the fungus was growing, the staphylococcus was inhibited. He identified the mold's antibacterial substance and he named it penicillin. There is a huge class of antibacterial drugs that act in the same manner as penicillin, which are collectively called the beta lactams. These antibacterial drugs inhibit the enzyme responsible for cross-linking peptidoglycan during cell wall synthesis. And in addition, we have something called glycopeptides. And an example of this is vancomycin. And vancomycin is able to bind to protein components of peptidoglycan and prevent them from being incorporated. This image here shows how peptidoglycan is normally synthesized. You have your chains of NAG and NAM that are alternating with each other, and then the peptide short chains of amino acids called peptides are built in as bridges that connect those different NAM subunits. And as the cell grows, it needs to build out a larger and larger cell wall. And there are enzymes that are responsible for catalyzing the peptide cross linkage. Now, when a beta lactam, such as penicillin, is introduced to the cell, these beta lactams, they interfere with those linking enzymes of the NAM subunits. And the cell has to continue growing or repairing its cell wall as time goes on. And if it was to continue to grow, for example, we can see that the cell wall is not rigid enough to prevent that plasma membrane from bursting open due to osmotic pressure of the solutes inside of the cell. So essentially, this cell can't maintain its cell shape, and you have a plasmolysis that occurs there. Now, there's a lot of different antibiotics out there that piggyback on this system. You don't have to memorize all of the different antibiotics listed here and know how many members, rings that are linked to the beta lactam there are, but you should be just understanding that here's our beta lactam. What this is a carbon skeletal structure, as we've described before. That means each point here is a carbon. And if it's not written, there's implied that there's hydrogens bound to these carbons as well. And so this is a four-membered ring, this beta lactam, with one being a nitrogen. And this beta lactam is what's responsible for interfering with that enzyme. And we can attach all sorts of different side molecules here, whether they are naturally occurring, semi-synthetic, or synthetic, that maybe evade the ability of that microorganism to combat these particular antibiotics. So with each antibiotic, that organism might have to have a specific new gene in order to dismantle it, or another genetic modification. So it's kind of this arms race where we're trying to develop new antibiotics to attack the same target. So again, as mentioned, the beta lactam antibiotics contain that beta lactam ring, which is essential for that function. And if the beta lactam ring is broken, the drug becomes useless. So bacteria come up with these beta lactamases. These are enzymes that break that beta lactam, and this can lead to resistance. And this is such a big problem, actually, that sometimes we introduce those antibiotics combined now with beta lactamase inhibitors that go after those beta lactamase enzymes. Mycobacteria, such as those that cause tuberculosis, have a waxy cell wall that contains a chemical we call mycolic acids. These are waxes that are present on the cell wall. And we have now a new target that we can go after. And mycobacteria are, because of those waxy outer surfaces in their cell wall that they contain, they're really hard to penetrate with drugs in order to attack other targets in the cell wall or within the cell. But we can go straight after the mycolic acids themselves. And one of those is a isoniazid, which inhibits the synthesis of mycolic acids. And another is an ethambutol, which prevents mycolic acids from being incorporated into the cell wall. These drugs, therefore, are really important in being able to treat tuberculosis. Drugs like polymyxins disrupt bacterial cell membranes. They do so by binding to phospholipids, which leads to leakage of cell content. So when we say we disrupt it, imagine those phospholipids being organized in that fluid mosaic model. And when this polymyxin binds to those phospholipids, it can create a gap in that lipid membrane, and therefore it's no longer a semi-permeable membrane. It's just a permeable membrane, which leaks cell contents out, which you can imagine that's a big problem, as the chief function of the cell membrane is to sort of demarcate what's not the self and what is the self. And this particular treatment is mainly used for gram-negative species. It is toxic when used systemically. It can go after our own cell membranes. And so we typically don't use it as like an oral dose or as an IV or intramuscular. Rather, it's typically applied topically. In fact, you may be familiar with the ointment Neosporin or other non name brand topical treatments for cuts or injuries that you might have. And as you look at the ingredient label here, you're going to see that it has polymyxin present on the label here. With a few other antibiotics, it's more effective if you use multiple antibiotics all together and kind of cover all of your bases. And because you're not really exposing yourself systemically, there's less of an issue of causing toxicity to the patient. But one thing that I should point out is that you don't want to use Neosporin on a very large area of skin or very deep wounds, because again, this can target our own cells. And remember, the idea is we want to try and target the disease-causing agent, not our own cells, as best as we can. To the left is an image comparing the prokaryotic and eukaryotic ribosomes. And recall, prokaryotes have that 70S size versus the 80S size found in eukaryotes. And additionally, they are made of subunits with their own sizes. You'll note that the prokaryotic one has the larger 50S subunit and the smaller 30S subunits. And some antibiotics target bacterial ribosomes, preventing protein synthesis. And because all bacterium have ribosomes, and they are relatively similar in function and form, antibiotics that attack these ribosomes are generally considered broad-spectrum drugs. For example, the macrolides, in particular, there are several of these, but azithromycin is the most common. They actually bind to that larger 50S subunit. Try and remember that, because macro means large, micro means small. So macrolide binding to that larger 50S ribosomal subunit. We can also inhibit the small subunit. There are the aminoglycosides. This includes the very common streptomycin and gentamicin and neomycin, but they bind to the 30S ribosomal subunit. Tetracyclines bind to both subunits, and this makes them sort of a very broad spectrum antibiotic. So here we see the macrolide, the azithromycin, as I gave you an example. And you can see that what they do is they're going to stop the synthesis of a protein. So here we have our mRNA. We're showing the 50S portion only. And this is going to blind and block the proper mRNA movement through that ribosome. And if you cannot move the RNA through the ribosome, then you can no longer read the code within the ribosome and create the proper amino acids shown with these grayish colored balls here. And then we have, this is supposed to represent the tRNA and the anticodon. So this azithromycin stops the movement of mRNA. There are aminoglycosides. They're sometimes still used, although less common. And what they do is they cause a change in the 30S shape. And if you cause the 30S shape to be misshapen, then you don't get proper binding between the codon of the mRNA and the anticodon of the tRNA. And so you get misreading that occurs. And so instead of adding the correct amino acid shown with these grayish circles here, you're going to get the incorrect ones shown with these greenish circles here. And those are your aminoglycosides. Tetracycline is a very common antibiotic, and as you can see here, we're focusing on its effect on the 50S subunit. So this is where the tRNA's dock is on the 50S side. And tetracycline has a pretty straightforward way of interfering with amino acid synthesis. It blocks the docking site of the tRNA. tRNA can't bind, it can't read the codon with the anticodon, and so it can't figure out which amino acid to put down. So it doesn't stop the movement of mRNA, as we saw earlier, but rather it just blocks the site, and that prevents the elongation of the amino acid. And finally, we have the oxazolidinones, and these are typically used for MRSA and VRE infections. We'll talk about that again briefly in the future, but MRSA just stands for methicillin-resistant Staphylococcus aureus. And VRE stands for vancomycin-resistant Enterococcus. And so these particular microbes are resistant to some of our more strong antibiotics. And so this one might be pulled out to be used for that sort of a situation. So this particular antibiotic works pretty simply, and it prevents those two subunits from coming together. What happens is you have your start codon, that methionine, that ATG or AUG present on the mRNA, and that signals that the two subunits should come together, clasp together, and start creating your amino acid sequence for your protein that needs to be synthesized. And oxazolidinone just simply attaches to the mRNA there, the 30S structure, and prevents the 50S from being able to clamp down. As we saw with protein synthesis, we can target the DNA and RNA synthesis of bacteria as well. Rifamycin specifically binds to RNA polymerase and prevents RNA synthesis. And this can be used in order to treat tuberculosis or traveler's diarrhea. And then ciprofloxacin inhibits the bacterial DNA gyrase. And this gyrase, we won't get into too much detail here, but bacteria don't package their DNA with histones, but they do like to create these, what we call super coils that are introduced to it. So they basically create it, kind of take the circle and imagine you twist it up, and they'll do that. And this particular ciprofloxacin interferes with DNA gyrase from doing its job, as well as something called topoisomerase. And topoisomerase, if you imagine this circle of DNA, which is the form that bacterial DNA comes in, sometimes when the cell machinery makes a copy of DNA in order to make two daughter cells through binary fission, what ends up happening is they get linked together. So if you imagine like chainmail, or if you take your finger and your thumb and you latch it over the other finger and the thumb, you make two loops that are attached to each other. Topoisomerase can cut the DNA and reattach it, separating those two loops so they're two free circles again. And ciprofloxacin acts on that activity. This is a really good antibiotic for treating UTIs or, again, traveler's diarrhea. Some bacteria synthesize their own nucleotides, and they do so using a starting ingredient called para-aminobenzoic acid, or PABA. And we have these drugs called sulfonamides, sometimes just referred to as short sulfa drugs. And these, the way they work is they mimic PABA. So they look very similar to it. And by mimicking it, they can block nucleotide synthesis. So synthesis of DNA, ATP, RNA. And here you can see how this sort of works. So by sulfonamide mimicking that, well, these enzymes try and act on sulfonamide, but they unfortunately cannot. And so this does something that we call competitive inhibition. It's competing with PABA in order to inhibit those enzymes that use PABA to make folic acid. And we can see that these sulfa drugs, they all have a similar pattern as we discussed with beta-lactams, having a key component to it here. We can see that this key component here is this cyclohexane ring here attached to it being an amine group. And it's very similar in structure to this para-aminobenzoic acid. Okay, so that's enough coverage for antibacterial drugs. We're going to briefly touch on all of the rest now, since they are less of a issue in a hospital setting than bacterial infections. The first up is antifungal drugs. And fungi are really hard to treat due to their similarity to humans. They are eukaryotic in nature. And if you go back and look at the three domains of life, we're right next to each other on the tree of life. One of the targets that we can go after is this lipid found in our membranes. And in our membranes, we have something called cholesterol. And in fungi, they have a relative of cholesterol called ergosterol. And antifungal drugs can attack ergosterol, and they can also attack the chitin, which is present in their cell walls. And again, we do not have cell walls, and we do not have chitin in our cell walls. And this just shows you the way this works. So these can be plasma membrane disruptors. So they bind to that lipid called ergosterol. We can see it sitting here in the phospholipid bilayer of the fungi. And when it does bind to it, here's this amphotericin B, it causes a channel or a pore to be created. And just as we saw with the polymyxins, you can imagine if we have a pore, that means that there's no longer a intact membrane. This disruption allows things to go inside or outside of the cell. This can be toxic to humans because human plasma membranes are also affected, although not to such a great degree. but there's not as big of a difference in targeting as we saw with something, say, going after peptidoglycan synthesis. Fungi again are really hard to treat. So let's go ahead and take a look at the antiviral drugs. Viruses are challenging to treat because while they are very different than humans, they use our own cellular machinery in order to run their metabolism and reproduce. Our common antiviral drugs that we do have are acyclovir and azithromycin and oseltamivir. And these, specifically the acyclovir and the azithromycin, these interfere with nucleotide replication. And while we do nucleotide replication too, we reproduce, we do DNA replication, we do RNA synthesis called transcription. These particular ones have a much higher affinity for viral enzymes specifically. So let's go ahead and take a look at these then. Acyclovir is and it goes by the brand name you might see as Zovirax, but it mimics guanosine. Guanosine again is one of the 4 letters in the DNA or RNA code, and it specifically goes after viral replication. But you can see the similarity between guanosine and acyclovir here. And by imitating it, if this gets incorporated by the viral enzyme responsible for DNA synthesis, it can actually cause premature termination of that synthesis of the strand. Oseltamivir, you might hear the brand name Tamiflu. This blocks neuraminidase, or NA as it is often abbreviated, or N in this image here. And this prevents the flu virus from being able to escape the host cell. Remember, this neuraminidase helps degrade the cell membrane and allows the new fully formed virion particles to exit the cell. By doing so, you can inhibit the spread. And of course, that's going to lessen the symptoms and shorten the duration of the illness. Now, azithromycin, this is another analog. It's a thymine analog. And this particular analog is more specific for that reverse transcriptase enzyme in our viruses that are called retroviruses, that are able to convert their DNA into, their RNA into DNA, the opposite process of what we see in our own cells where we transcribe DNA to RNA. So it's called reverse transcriptase. One of those particular retroviruses that this is particularly effective for treating is HIV, human immunodeficiency virus. Again, the selective toxicity is difficult to treat, so these drugs, they often have some side effects, but this one works pretty darn good. All right, moving right along, let's talk about the anti-protozoan drugs. Protozoan infections, protozoans are eukaryotic, so making unique targets is difficult, and therefore treatment is really tricky with these ones. Quinolines, they prevent protozoa from breaking down hemoglobin, and this is useful in treating malaria. And then we have pentamidine, which binds to DNA in a protozoan organelle called the kinetoplast. It is effective for African sleeping sickness. And these drugs target unique features of protozoa while trying to minimize the toxicity to human cells. So quinolines and pentamidine. And finally, we have the anti-helminthic drugs. Helminths, again, are parasitic worms. They have complex life cycles, and they, in many ways, resemble human physiology, which again makes these guys difficult for drug treatment. We have a couple that we're going to talk about here. One is ivermectin. This blocks worm-specific nerve transmissions, which hopefully causes paralysis in those parasitic worms and death. And then the next one is praziquantel, which includes a chemical that induces calcium influx. In other words, it causes the cell to take in a bunch of calcium, and that can cause paralysis in those worms and hopefully death as well. You might recall hookworms from our eukaryotes of microbiology chapter. They have a prevalence of about 500 million people infected with them globally. And that's a pretty big number. That's greater than the total US population. As with other helminthic drugs, it is tricky to find a target that is specific for the worms but not to humans. Tubulin has a slightly different amino acid sequence in worms than in humans. And we talked about tubulin when we talked about the cell in chapter 3. And tubulin is really important for example, it's involved in the centromere and positioning of the centromere. And it's made up of amino acids that auto-assimilate, and they're a little bit different in sequence in worms. And so we can attack that difference there. And albendazole disrupts that microtubule formation in worms. It also has some action in preventing glucose uptake, which of course is needed for energy production. So that covers the main categories of antibiotics. Now let's talk a little bit more about drug resistance, as it's becoming more and more of a problem in healthcare. Antibiotic resistance emerges through genetic mutations or gene transfer between organisms, and it is driven by the selective pressure of antibiotics. For instance, mutations in ribosomal proteins can slightly alter their structure, and it renders an antibiotic maybe ineffective against that ribosome in that particular way. And in a microbial population, a small fraction may naturally possess that genetic modification that allows it to be resistant to that antibiotic. And so once exposed to the antibiotic, you clear out the microbes that are not resistant, leaving the ones that are resistant. And this leaves them to have this favorable environment for both survival against that antibiotic, but also proliferation, because there are no longer other bacterium within that population competing for resources and space. Okay, so we've talked about the different antibiotics and their different targets that we use for antibiotics. And I don't want you to get this confused with targets. These are different mechanisms for drug resistance. So these are ways that microbes circumvent antibiotics. And these include 4 different ways, broadly speaking. One is blocking drug penetration or rapid efflux of that drug. So you can have, as you can see here, this is a pump. And the idea is that this pump can rapidly pump out whatever antibiotic that's going into that organism. Or maybe you just block it from entering altogether. So maybe you change the shape of a porin, which would usually allow that antibiotic to get in. You change the structure of that protein, and you can no longer get that antibiotic into that organism. A great example of this are the mycolic acids of Mycobacterium. Those mycolic acids create that waxy layer that surrounds the cell and makes it difficult for those antibiotics to penetrate into that organism. So blocking penetration or uptake of drug, or rapidly pumping out the drug, is one way to do it. Drug inactivation is another. So you can break down that drug before it can act. So we talked about the beta-lactamases, and those break down beta-lactam antibiotics. And so organisms might have some sort of an enzyme that allows them to quickly degrade that antibiotic before much harm can be done within the cell. The third method is target modification. This is where you alter the structure, which is the target. Can be done through mutations over time. or the exchange of genetic information between different microbes. The example that I gave earlier was, imagine that you have a particular antibiotic that attacks one of the 30S or 50S ribosome units, and over time, that ribosome can evolve and change its structure and amino acid sequence, and then that drug might not be able to recognize it anymore or bind to it or interfere with the process. And that is target modification. And the final method is, well, maybe you're not going to modify that target, but you're going to produce a lot more in excess. And so that's called target overproduction. So maybe, let's say you have an analog that mimics PABA, right? Your sulfa drugs. And this organism, instead of, it can't really change PABA, which is just important starting ingredient or ingredient in order to make your folic acid. So what it does is it just makes maybe a lot of PABA. And so even though you're providing something that mimics it, that competitive inhibition is overridden by producing a lot more of that compound that the cell needs. Some bacteria develop multiple resistance mechanisms, making them multi-drug resistant superbugs, or MDR. These infections are responsible for greater than 2 million cases and 23,000 deaths annually in the US. And examples of this include MRSA, which is, again, the methicillin-resistant Staphylococcus aureus, and our VRE, vancomycin-resistant enterococci. We also have the CRE, or the carbapenem-resistant Enterobacteriaceae. These infections are often untreatable with standard antibiotics, requiring stronger or a combination of antibiotic therapies. And lastly, we have pan-resistant CRE. This is, again, carbapenem-resistant Enterobacterialis. This refers to a group of Enterobacterialis bacteria. And Enterobacterialis bacteria, this includes our enterobacteria that we find in our digestive systems and our guts, such as Klebsiella pneumoniae and Escherichia coli. And these pan-resistant bacterium, what that means is that they're resistant to all available antibiotics, including the carbapenems, the beta-lactams, and the non-beta-lactams that we discussed, such as the aminoglycosides and the polymyxins, et cetera, et cetera. Now, I just wanted to end on a note of what's causing this antibiotic resistance. We discussed the mechanisms, but what is causing this? What is driving this in our current day? Well, one is the overuse and misuse of antibiotics. In some countries, antibiotics are available without prescription. And even when prescribed, they are often given for viral infections, which they cannot treat, or given for the wrong type of infection. So you might be giving someone an infection for a drug that's narrow spectrum for gram-positive bacterium when there's a gram-negative infection. And why is that an issue? Well, you're exposing a lot more, you're giving a lot more exposure or natural selection to these bacteria, to these antibiotics. And the more exposure you give, that small percentage of the population that is resistant to them is going to thrive under those conditions and become more prevalent. Additionally, we're seeing insufficient dose regimens. So if individuals or doctors who prescribe them, they stop antibiotics too early, that allows those microbes that might be resistant, and some of them may not be completely resistant, like we discussed with our different mechanisms. Let's say a bacteria is producing just more PABA. Well, if you're producing more PABA, that doesn't mean you're completely resistant to a sulfa drug that mimics PABA, but enough to survive the length of the course that was provided. So bacteria that may have partial resistance to that antibiotic, if you don't give enough antibiotic for long enough, it may survive and then it may mutate further to become even better at what it was doing. And the last one you might find surprising, and that is livestock supplementation. Shockingly, the numbers have gone down and we are getting better at this, but about 70% of US antibiotics are used in animal feed. This is mostly tetracyclines, and these are low dose exposures that select for resistant bacteria, which can then spread to humans. We use these in animal feeds because it can allow and encourage for animals to grow bigger and healthier if you're fighting off bacteria or other organisms as well. And so it's really hard to get off of this usage of these antibiotics. And yeah, it's a big problem in the United States. But that wraps up our discussion here of antimicrobials. Hopefully you enjoyed, and please join me in the next one.